Uh, Thank you, Prashant. Are you being able to see the slide? And I hope it is changing also. Yeah. It didn't change. Okay, let me look at that. Yeah, I'll one. Yeah, I have this problem sometimes with Google, and yeah, so there's a way to handle this, and let me see. I'll just see if it doesn't work. I'll not go in the presentation mode. I'll just go then in this mode, and is it changing now? Yeah. No, this is a problem. So I think I'll keep it like this. Otherwise, it will take time. Uh, Thank you, Prashant, for the introduction and giving us an opportunity to talk to an audience of people from bioinformatics. And so, now this is a talk that I work for a company called TissueGnostics, and as you can see, it's about tissues. And here we do the analysis of tissues by imaging. So, and I therefore had to think that how do I connect with the audience here, and how much is our work actually related to informatics? Uh, Although interestingly, the subject is related to the upcoming subject of bioimage informatics. But if you look at it in the informatics domain, it actually occupies, I think, a peripheral position. So let me just introduce to you Bioimage Informatics. What is it about? And also the technology that I'm going to the talk that I'm going to give to you today is about a technical approach that the company is taking called TissueFacts in relation to Bioimage Informatics, and what are the outcomes of that. I'm sorry. So, what is bioimage informatics? Bioimage informatics is a subfield of bioinformatics and computational biology. It focuses on the use of computational techniques to analyze bioimages. And especially these are cellular and molecular images at large scale and high throughput. So this is what we exactly do, that we what we have is our images of tissues, and these are therefore not at the subcellular level mainly. These are at the cellular level, to a certain extent subcellular. They are large scale because we handle images of large tissues in this technical approach, and they're high throughput because there is a high throughput of data that because we have to acquire data from a large specimen, and also that if you are doing, let's say, a population study, you would like to do you would like to look at many sections, and therefore the number of specimens will be more. So you need high throughput, as you can imagine, high throughput imaging, high throughput microscopy for that. Now, this is a subject which has of bioimage informatics, which has been building up since the last 15 years, roughly. And 2005, people started talking about it. And in 2010, the ISMB conference had a session on bioimaging and data visualization. And then around the 2010s, bioinformatics and BMC bioinformatics, they started introducing bioimage informatics as a topic for their journals. And lately, the European Union for EMBL has been very active in this field, and it also has set up an archive where people can actually upload annotated images for informatic analysis. To give you a view of what currently bioimage informatics is about. This is a screenshot from a conference in last year organized by Institute Pasteur. And as you can see, the topics that were discussed here is advanced analytical solutions for bioimage processing and analysis, that you would actually have to process the image to make it analyzable and then do subsequent analysis. Statistical spatial analysis. Now, statistical analysis is a part of informatics of cellular and molecular distributions. This is exactly what we do in our work. Applications of machine and deep learning to analyze cellular structures and related functions. I mean, this you can very well relate to. Quantification of dynamic images and transport phenomena. Now, by dynamic images, we mean that when you are looking at an image which is changing, and typically when it comes to microscopy, it's about live cell imaging or live tissue imaging. We are also involved in that. I won't be able to discuss much of that because of time. Dynamic cell imaging, exactly, for live cell imaging. Reconstruction and analysis of structure and function of biological networks. Here we do, what we're looking at is the interaction of the organization of cellular networks, interacting cellular networks, and their organizations between subpopulations of cells. Registration, correlation, and fusion of multimodality data. Uh, mm, uh, this is also another thing which we do. Multimodality, by meaning, is that can I have a cell level MS data, mass spectroscopic data, being integrating with antibody stain data, integrating with RNA omics data? Can I do this kind of multimodality DNA integration? And also, can I now actually integrate this with an H&E, with the data from an H&E stain section, where there is only morphology there and no molecules? So this is what it's basically about, and I think you have got an idea. But we also deal with one thing. We also deal with the subject of cytometry, and because this is an audience of very diverse, let me define what cytometry is about. Cytometry is about collecting data from one cell at a time for multiple parameters. Now, classically, this has been done, as you would know, for the very popular name of flow cytometry. But in flow, you don't collect images. Now, in order to do the same thing, what flow does on a single cell basis, for a section which is actually for a section of a tissue in which cells are not in suspension, as in blood. In flow cytometry, the cells are naturally, the native state is actually in suspension in blood. I mean, in suspension. But in a tissue, they're all organized, and therefore it would not be right to actually disturb this organization, make a suspension, and then do flow cytometry. There'll be two or three problems with that. You would be losing the spatial information of the distribution of cells, number one. Number two, you would change biology the moment you dissociate them. There would be short-term changes immediately happening, like changes in protein phosphorylation profiles. And thirdly, you would not be able to collect image data. I mean, all the flow cytometry is now coming up with imaging, but at the resolution at which it's possible by just doing microscopy imaging, let's say with a 60x or the 100x lens. So there is a subject of tissue cytometry here. And so therefore, what the company loves to call this is as tissue facts, which is basically using a flow cytometric language to understand the issues, the story of a tissue. And the data can come from sections which are either stained with non-fluorescent dyes, which will mean a low-cost, affair, where it is stained with H&E or with antibody dab, but because such staining will not allow you to look at many molecular targets, you can't multiplex. If you want to actually look at many molecular targets, you'll have to do a multi-antibody staining, and this doesn't allow that. And that is allowed, as you would understand, very nicely by fluorescence, and so one resorts to actually fluorescence staining. But both methods allow you a cytometric analysis, as you see in these pictures here. Anybody who's there from flow cytometry will immediately relate to this kind of data, which is very common in flow cytometry. Such data can also be obtained by tissue cytometry. Now, so cytometry is basically about, as I said, collecting data from one cell at a time. And in this today, we are talking about collecting image data. And the so we from the image data, we actually can find out populations and subpopulations of cells. Now the question is that we have come to understand, come to discover, let's say, populations and subpopulations. But where do all these cells or subpopulations lie in the overall context of the tissue? And that is about a more global spatial analysis that can be done. So it's a blend. So what the subject of tissue facts, it is actually a blend of single cell image based cytometry combined with spatial analysis and on a global scale, and a global scale of the tissue. So the now this has also become more important because with the coming up of the project called the Cell Atlas project. Many of you would have heard of this project called the Cell Atlas project, in which it is actually it is trying to identify all the different histological cell types in a tissue. And now if we look at the different histological cell types, we can and if we and where each dot here, for example, represents a single cell, we find there can be different populations or subpopulations of cells. There will be a particular population subpopulation profile in a healthy individual, and then depending on the disease the healthy individual encounters, there can be population drifts. There can be drifts in the cell numbers, or there can be creation of new subpopulations of cells, or even for the same disease, depending on the course of the disease, there could be different population changes, and these population changes could be affected for different reasons. They could be affected for a change in cell-cell interactions. It could be because of, let's say, a new phenotype appearing, for example. Going further, so supposing we want to therefore now do actually tissue cytometry, and in this case we are actually doing, I mean, let's more or less we can actually call it, we are doing image informatics of a tissue. So the first thing is to actually therefore collect the raw data. The raw data will be collected, because then for that you'll have to stain the tissue. You'll have to stain the tissue for different things, and therefore it will be multiplexed imaging, staining and multiplexed imaging. And after the imaging, you would have identified different cell types, and now cytometrically you can now actually place them, you can do statistical analysis on this and find out the different populations and subpopulations that are there. And once you have a subpopulation, you would like to see that this subpopulation or a single cell from that subpopulation, when it is placed in the context of the tissue, what are its immediate neighbors like? How far it is away from the neighbors? A typical example would be, let's say, an infiltrating in a tumor, an infiltrating leukocyte interacting with the tumor cell. The closer it is to the tumor cell, the more lethal it is supposed to be. And that's the data which has been observed from the clinical analysis. And so therefore, the physical proximity measurements correlate with lethality. So this kind of spatial analysis can be done. So how does the workflow look like? The workflow therefore naturally has to begin with multiplexing, and then by whole slide imaging. Whole slide imaging, because we have to collect data from a large section. The bigger the section, the richer the data. And because we are also looking at single cells and collecting data on a cell-by-cell basis, if there is a small subpopulation of cells, which is around, let's say, 50 cells in a whole section of 300,000 cells, it will have to be collected on this. You'll have to scan a large section to find these few cells in number. I mean, cells which are few in number. So you do whole slide imaging. After the whole slide imaging, the imaging is done. You using the software, you identify single cells in the entire section, and you find single cells of, and depending on the molecular markers that you've used, you find actually single cells of different types, of different histological types. You do then a cytometric analysis. You also do a tissue classification. This is again, this is AI based. I won't have time to discuss this. And now, so the tissue classification tells you that in the tissue, which are the global histological distributions of cells. If it is skin, where is my epidermis and where is my dermis? And in that global tissue distribution, where do my subpopulations lie? And that is, and if you combine these two, it's what results is what it results in can be called as spatial analysis. So, and this is what is, so this is the basic data collection is done by what is called as whole slide imaging. The whole slide imaging involves having a slide with a large section, and the image is actually, and the tissue is scanned to collect data from the entire section. As you can see in this image here, this is an image of a colon cancer specimen, in which the whole section has been scanned. You find different cells, subpopulations of cells, and although the section is large, you can actually collect data to make it computationally less intensive from small regions of interest. So you can have regions of interest of different sizes, and you can collect data from that. But if you will get the most reliable data naturally, if you actually look at the entire section, or you have an ROI as big as this section is. Now let's take a few examples. So here we have an example of a tumor tissue being infiltrated with leukocytes. So we have all the nuclei in the tissue. I mean, actually, all the cells and their nuclei stained by using a nucleus-specific stain called DAPI. And the infiltrating lymphocytes are of two categories: the ones having a CD4 marker, another having a CD8 marker, and another having the FOXP3 marker. The FOXP3 is a transcription factor. The CD4, CD8 are cell surface markers. Now, the first job will be we have to identify single cells in this, and this is aided by the fact that all the nuclei in all the cells is stained with DAPI. It is. So how does this actually help? So the nucleus seeding is taken as the seed, and based on that, we identify what is the cell morphology like. I'm sorry, I won't be able to show you here the full the slide in the presentation mode, and that shows how the algorithm works. Here, what is done is that the nucleus staining is done, and then the algorithm proceeds to look from the nucleus onwards towards the cytoplasm and towards the membrane periphery, and looks for signals. And as it goes, it keeps on looking for signals, and then it hits the membrane boundary and then stops. And therefore, it can actually make out the true morphology of the cell. So this way, this way delineates the morphology of the cell, and that's how it delineates it, and it tells it that is a single cell. So this is a cell stained with one particular marker, this is with another marker, and that's how the cells are stained for different markers. I'm sorry. And therefore you can do actually a cell count based on the types of markers there. So we'll say, for example, in this tissue, there are two kinds of cells, or the green cells and the red cells. So now we come back to our slide, and we find that we have now identified the different cell types based on their DNA staining. We know that how many total number of cells are there, and of these total number of cells, how many cells are, for example, CD4 positive or CD8 positive or FOXP3 positive. Now, this is a data which is taken from cells. We are trying to look at co-expressors. So this is a plot of CD4 versus FOXP3. So we find that of all the cells here in this section, or in this field of view, there are some cells which are pure FOXP3 expressing, only express FOXP3. Here there are some cells which only express CD4, but there are some cells which express both, and these are these co-expressors. Now, each dot in this plot is coming and correlating with a single cell in the image. And here, and we know that these are co-expressors. Each dot, and that is each cell, is also expressing CD4 as well as FOXP3. And when all, okay, the other thing is that all the cells in this box, they are marked by this red boundary. Okay, so we have, what we have done is gated this population, and we see the gated populations marked by cells having red boundaries, and each of these cells with the red boundary has both CD4 and CD8 in it. Now we also see that some of these cells excuse me, some of these cells are also are yellowing. uh, of the CD4 and the FOXP3, the CD4 is a red dye. The FOXP3 is stained with a green dye. And if green and red are present in more or less equal proportions, we see a marked clear yellowing. Otherwise, also it's a yellowing, but it will be a different shade of yellow. Now, let's take a little bigger section and a bigger picture. Here we have again this section from colon cancer, and here we have stained this section for DAPI, because we need to actually identify all the cells here, and the DAPI stains all the nuclei and therefore helps enable us to actually identify all the cells. Then we have labeled it with cytokeratin, antibody labeled to cytokeratin. Cytokeratin will label all tissues here, and therefore also the tumor tissue, because this is a colon carcinoma. And then we have the infiltrating leukocytes being stained by CD4 and FOXP3 as in the last slide. And then we have Ki67. It's a nuclear marker for dividing cells, for mitotic cells. Now, our first job is to identify all the nuclei, and to identify all the tumor regions. And here we have a picture where all the nuclei, including the epithelial nuclei and all the nuclei in the tumor stroma, are covered by this gray or greenish gray cell nuclei that you see here. And the cytokeratin is shown by this yellow or orangish mask. And we have combined two masks here, the nuclear mask versus the cytokeratin mask. And so the cytokeratin here is the tumor tissue. Now the question is, we want to identify that there are leukocytes and leukocytes of different populations in the stroma, and some have infiltrated into the tumor tissue. We want to identify that how many of these have entered into the tumor, number one, and does the number distribution of a particular population of leukocytes, does it correlate with how far it is from the tumor? Does it have a spatial correlate to it? So what we do is we actually take the CD4 positive population and we gate it according to the distance from the, go back here, we gate it according to the distance from the tumor margin. And coming back to the slide here, so we gate it and we find that as we move, this is, we gate it for every 25 microns. So this is 0 to 25, 25 to 50, and this way we go up and we come to actually around up to 250 to 300 microns. And we find that as we move away from the tumor margin, this is closest to the tumor margin, as we move away, we find that the CD4 population is maximum near the tumor margin, and it actually, in a very monotonic way, it actually decays, the numbers decrease. And these numbers are reflected here in this table here. I'm sorry, not being able to show you in the full slide, but these numbers are reflected here, as you see in the graph here. So now what we can do is that I want to now picturize, I want to visualize the number of CD4 leukocytes which are present at 1 to 25 microns from the tumor margin. And these are visualized in red here. Okay. So we see the distribution of that. And then we come across. Now what I would like to do is I would first of all like to do two things. I would like to visualize the numbers of CD4 here according to distance, and the distance, as I showed you in the last dot plot histogram also. I see that curve here. And then further, I can now look at different subpopulations. I can look at the cytotoxic T cells, the effector cells also, as they as a function of the distance from the tumor margin. We get this kind of distribution here. And if you're looking at one particular what you call CD4 only, for example, this is a picture that you are seeing here based on the distribution of the one particular subpopulation, the CD4 population, in which the ones which are the green ones are the ones which are actually present at 0 to 25 microns, and the ones which are the violet are placed farthest away, and they are at 250 to 300 microns. So I gave you an idea of the possibilities of image analysis. A little note towards the end of the kind of hardwares that are possible. Now, you can begin with a very basic hardware, uh, which will actually only acquire data from chromogen stained images, not fluorescent. And the hardware complexity goes up depending on how much data and what rich, if you want more detailed data and richer data. So the next step is, if you collect fluorescence data, then you have another model. If you want to go beyond fluorescence, if you want to collect spectral data, even up to a resolution of 10 nanometers for a wavelength range of, let's say, 400 plus to 700 plus nanometers, then there is a spectral imaging device which comes fitted with the whole slide imager. Also, you can fit it along with the confocal. Now, what a confocal microscope allows you is that it allows you to look at a thicker section. And a thicker section is important because it will allow you to collect data from more cells. A confocal basically allows you to look at thick sections, and to be able to visualize, to be able to identify and visualize single cells there and collect data. And as I told you, this is essentially about looking at data on a single cell basis from populations and subpopulations. And there could be some subpopulations which are very small in size. They could be composed of only, let's say, 200 cells, and therefore it will help actually if we collect data from a thicker section, and therefore our data will be more statistically reliable. Now, also, this is about informatics on a bigger scale. That means if you are doing a population cohort study, then you would need to actually run multiple slides. Okay, assuming that one slide is coming from one person or one patient, and you would like to run multiple slides. And so the standard microscope, which you see here in this picture here, it can accommodate 8 slides only. Good for a lab setup. But if you want to do a cohort study, then you would like to actually feed in more slides, and then this slide loader can automatically keep on loading 120 slides one after the other. And you can therefore do a cohort study for a cohort size of 120 individuals. A little word before I leave you about the company. It's an Austrian company, and as you can see here, this is a nice old building from Austria where the company is headquartered in Vienna. It was started in 2003. It's now over 17 years old. It was started by Dr. Rupert Ecker. He himself is a scientist, and he himself made one of the very seminal publications of being able to do such data collection from solid tissues. That paper is highly cited in reviews, and that and gives an idea of what kind of the importance of that kind of work is. We, as you can also see, I mean, we do bioimage informatics, but a lot of the work is also to do with disease tissue. Therefore, this is a subject which is very much connected and very much a part of digital pathology, as many of you would know. And also, there is a website in which all the publications that have used papers, that have used products from the company are listed there. Currently, it's around 2,000 publications have come up which have used this data. This data is can be filtered there online based on tissue type, based on research area. So, if you're interested, you can actually you can go there. Thank you. Yeah. So what has happened is, and this is the way things go happen actually, is that uh, there has been this omics, transcriptomics data which has been driving this a lot. Okay, and this started off for historical reasons. But what has been observed is that, especially for example, because you said translational, in tumor in immuno-oncology, it is seen that there is a very high correlation uh, between antibody-based protein data and cell types than it is with using it with transcriptomics or even with DNA omics. This is very well established, very early studies, but this is getting so. Uh, so what is becoming important in immuno-oncology, in order to find out whether a particular drug will work or not? Number one, to find out the correlate. Number two, whether it will be a person will be a responder or a non-responder. In this spatial analysis is becoming more important than actually knowing the expression data. So it's a combination of expression data along with the spatial distribution, which is becoming more important. The spatial analysis is becoming more important here than in other fields. So wherever you look for in immuno-oncology, it's all about spatial analysis. I think, yeah, if you buy the whole system, because although it is based on a microscope, but the first common question is that you say, I have a microscope. Can I just buy your image analysis stuff, right? Now it doesn't it works and doesn't work really that way. uh, it does not work that way completely, because it is very important, because a lot of the algorithms now are being driven by AI, and here the you need to have a control of the variation of the data, of the input raw data. And for that again, for more precise what you call algorithm to work, you need to have a high precision microscope. So now what has been zeroed upon is that the best microscope that works for this is the Zeiss microscope, number one. Now, number two is so therefore that restricts other microscopes. Other microscopes, some microscopes have been allowed, not all, in which it has been tested. And then again, the other thing is that you not only need the image data, you also need the metadata, because you will have to rebuild the whole tissue. Because if you just have the image data, you don't have the data of, because the image is acquired on per FOV, and for each FOV you need to know what are the X and Y coordinates and what is Z coordinate, and what is my camera data for that particular FOV. So you need the metadata also. And so it becomes complex actually to actually incorporate the entire metadata along with the image file for open it up completely. So it is going to be restricted, yeah. Thank you.
Thank you, Parash, for giving me